Our final speaker for today is Professor John Bowman, who joins us from the School of Biological Sciences. After obtaining a Bachelor of Science in Biochemistry from the University of Illinois, Professor John Bowman trained as a geneticist during his PhD at Caltech, where he started working on the Arabidopsis as a model plant to address fundamental questions in plant development. During his PhD and subsequent postdoc at Monash, he contributed substantially to our understanding of the developmental genetics of flowers. In 1995, he established his own laboratory at the University of California in Davis, where his group discovered several gene families whose action determines the polar growth of leaves. During his time at Davis, John was named a Chancellor's Fellow and received a Beckham Young Investigator Award. In 2006, John moved as a Federation Fellow to Monash, where his laboratory continues to investigate fundamental questions in land-plant development with an evolutionary perspective. This year, John was elected a Fellow of the Australian Academy of Sciences. In his spare time, John is a guide at the Fairy Penguin Colony on the St Kilda Breakwater, and he also studies the colony with Earthcare St Kilda to work out how these birds coexist within a city of 4 million people. Today, John is going to talk on evolutions in the cycles of life. Thanks, John.
Thanks, Kay, and thanks for the little opportunity to continue sort of a theme of evolution, maybe slowly getting closer to us. I'll tell you a little story that actually all the work was done here at Monash in the last, under the auspices of the fellowship that I got, and I'll say that it probably couldn't have been done under a single grant cycle, so that was an argument for long-term funding of science. So when we look at life on Earth, multicellularity, like our bodies, evolved multiple times independently. So we're over here, multicellularity evolved independently in fungi, which we're quite closely related to in the big scheme of life, in brown algae, which is completely different. Well, oftentimes people think about brown algae as plants, but they're not really plants. They're actually more closely related to the malarial parasite than they are to plants. And then these groups are traditionally what we think of as plants, where we have some red algae and a couple of different lineages of green algae, and these are land plants. And multicellularity evolved multiple times independently in all of these lineages, and my lab primarily works on these land plants. But if we think about where we all came from, it was probably a single cell. And the life cycle of our common ancestor between us and brown algae and plants had a life cycle that was most likely similar to that found in a lot of single-celled organisms still alive today. So this is a Chlamydomonas, a green algae, and they have they exist as single cells. This is a marine algae. And when nutrients are getting low, they decide, maybe we should mate. And they differentiate into different types of gametes called minus and plus. You can think about that in terms of us as being sperm and eggs. And then these two gametes fuse, and then they have a diploid phase. So there's a single set of chromosomes in here called haploid, and in here there's a diploid. Then after fertilization or fusion, this diploid cell will undergo a process called meiosis, which produces 4 haploid organisms again. So this is the simplest life cycle, maybe if you go back to your first year of biology. Now, this life cycle can be modified to be multicellular in several different ways. So in us, what's happened is that this diploid phase here undergoes something that's called mitoses, which are just cell divisions producing more identical cells, and each of them have 2 copies of the chromosomes. And in us, in the males and in the females, only a small subset of our cells of our body undergo this process called meiosis, and of course that produces eggs in the female and sperm in the male, and these are single cells. They get together through fertilization, same thing here, to produce another diploid organism, and then that grows up. Now, but this isn't the only way multicellularity has evolved. In some green algae, freshwater green algae, instead of the diploid phase becoming multicellular, the haploid phase evolved to become multicellular to begin with. So then you have a multicellular complex haploid phase. of only which a certain small number of cells will differentiate either as motile sperm or as, actually in this case, an enclosed egg. The sperm fertilizes the egg, forming this diploid cell, which basically just undergoes meiosis to produce more haploid organisms that swim around. Actually, in the early days, in the 1800s, when they first started seeing plants producing motile cells, they called them animalcules or plant animals, because plants weren't supposed to swim around. But they're quite common in most lineages of plants, actually. Now, some organisms have evolved multicellularity in both generations, and the way this has happened, at least in the case of land plants, is it probably evolved from an ancestor like this that had this complex multicellular haploid phase, and then this diploid phase also evolved to become multicellular. So this is complex multicellular, produces a large body, of which a subset of cells undergo meiosis to produce more haploid organisms, which grow into multicellular things, which then produce sperm and eggs to complete the life cycle. So if moss is a typical plant that is like this, and so if you're walking in the bush, you can think about this when you see the moss. These little green leafy things, those are the haploid phase of the life cycle. In other words, that's like our eggs or sperm. Whereas the brown sporocarp, as they're called, fruiting body, is actually the diploid. So that's like our major part of our body, right? So another way to think about a complex life cycle like this is if we had this life cycle, it would mean that we would produce sperm and eggs, but the sperm would develop into some multicellular being, and the eggs would develop into some multicellular being, and they'd go out to the pub and meet and mate, and we wouldn't, our diploid bodies may not even know about it, right? So they'd be completely separate. So we're interested in understanding how these life cycles are regulated, and some of the insight came from a paper from Ursula Goodenough's lab at Wash U, who was working on this organism Chlamydomonas, and they found that the minus gamete produces a protein called NOx, and the plus gamete produces a protein called BEL. And these proteins are transcription factors. In other words, they're proteins that regulate the gene expression of other genes. But the NOx protein and the BEL protein can't work on their own. Rather, they need to heterodimerize in order to bind to the DNA to activate gene expression. And so you can think about these gametes as being preloaded with proteins that only after the fusion of the gametes are those proteins able to heterodimerize and then activate gene expression. So you sort of preload your gamete so that as soon as it meets a partner, it can start developing the diploid phase of the life cycle. So it turns out that in Chlamydomona, the NOx and the BEL basically activate genes that are expressed only in this diploid phase, which eventually lead to meiosis. Now, in land plants, it becomes a little more complicated, but they also have NOx and BEL genes. And one of the major events leading to land plants was a duplication, so that now they have two different types of NOx protein. We just call them NOx1 and NOx2. And we believe that these are actually preloaded into the egg cell. So you can think about the egg cell being the same as the minus gamete here, and that the BEL proteins are preloaded into the sperm, so you can think about that being the same as the plus gamete here. And then when the sperm fertilizes the egg, that these different heterodimeric partners can form. A NOx1 with a BEL and a NOx2 with the BEL, and they can activate or repress gene expression in the diploid phase. So I'll tell you a little bit of data that we had on the NOx2 genes in moss, and it turns out that basically what I presented in the last slide is based on this data, that the NOx2 is actually expressed in the egg cell, consistent with it being preloaded into the gamete, but it's not expressed anywhere else in the haploid phase of the life cycle. However, it is expressed, this blue staining as gene expression, is expressed throughout in different cells and tissues of the diploid phase of the life cycle. Now, in this particular moss, we can go in and we can eliminate genes by homologous recombination, which means we just delete the piece of DNA encoding a gene. And so when Keiko Sakakibara, who was a postdoc in the lab, did this, she eliminated the two NOX2 genes here, and it turned out that rather than getting these nice little brown diploid fruiting bodies, the mutant never made them. However, when she extracted the mutant, she saw that they died when they were about this stage, and they often produced these little funny outgrowths here. And so she took those mutant and wild type embryos at that stage, and if you take a wild type embryo and just place it on a petri dish with some nutrient, it'll just sit there. However, if you took the mutant one and did the same thing, after about a week you sort of got some filamentous growth here, and after a couple weeks this looks pretty much like a wild type haploid phase. However, this came from the diploid cells here, so this is, and when you look at the number of chromosomes in here, it's definitely a diploid organism. And so one hypothesis about what this gene does is that when fertilization occurs and you form a zygote and you want to enter the diploid phase, in organisms that have complex development in the haploid phase, you want to make sure that this program never gets activated in this phase. So one, you have to activate the diploid genetic program, but you also need to repress the haploid genetic program. And so one role of the NOX2 genes in moss, at least, is to repress the haploid program during the diploid phase of the life cycle. Again, you can use a human analogy. In this case, it would be as if our bodies all of a sudden de-differentiated into a mass of eggs and sperm, right? Shifting from the diploid phase to the haploid phase. So because I quite limited time, the function of the NOX1 we think is actually to activate cell division and promote the building of the diploid body. And the NOX2 is to repress the haploid body in there. And one of our ideas right now is that one of these evolved from the other one, and we think that the one we think that acts to repress gene expression evolved from the activator. And we think that this is probably a very common theme in evolution of transcription factors, that you have a transcription factor that does something, the easiest thing to do is evolve one it does the opposite thing, right? Can still bind the same DNA sequence, but interacts with different partners. And we think that this gene duplication, producing these two, was really instrumental in the invasion of land by plants in general. And the reason why we think that is that we need to build a complex multicellular diploid body in order to be efficient to reproduce on land. Because if you don't think about flowering plants with pollen, all of the first land plants required aquatic fertilization. They had motile sperm that would swim around. And now they're on land, you can only rely on having an aquatic environment to create new progeny a few times during the year through heavy dew or flooding or whatever. And if each fertilization event only gave rise to 4 progeny, like it did back in these, then each fertilization would only be a little bit of progeny. And whereas if you made a complex multicellular diploid, you could get 1,000 cells or 10,000 cells all to undergo meiosis, and all of a sudden one fertilization event would give you tens to hundreds of thousands of progeny, and that would enable the plants to colonize the land even when they couldn't undergo their sexual cycle all the time. So we'll stop there because it's the natives might be restless out there.